screen. All right, everyone can see the slides?
Yeah.
You good? Okay, perfect. So good morning, guys. Thanks for rocking up at 10 a.m. We're going to get started with GI Phys. So if any questions come up, you feel free to type them in the chat, or you can private message one of the others if you're a little bit like nervous or whatever, and they'll let me know. But we'll get started. So the first concept is peristalsis, and this is like really important because basically the whole way that we move foods through the GI tract is like through peristalsis. And it's basically the coordinated contraction of two different types of muscles. So you have the circular smooth muscles, which Kevin mentioned on Friday, and these ones, they wrap circumferentially around the lumen of the gut so that when they contract, it like closes off the lumen, versus the longitudinal smooth muscles, which run up and down the gut tube. And so when they contract, it shortens the gut tube. So you can imagine if you've got a bolus of food, the circular muscle will contract and close off proximal to the food bolus so that it doesn't like reflux back up. And then the longitudinal muscle underneath contracts to shorten the tube and pull the food further down. And with that coordinated movement, you get peristalsis of the bolus down the gut tube. And because all of this is smooth muscle, it's going to be controlled by autonomic nerves. And we did get quite a few questions about autonomic innervation on Friday. So I put in a couple extra slides, which hopefully will help out a little bit. But starting off with some general principles, the presence of a food bolus, it gets detected by sensory neurons, specifically mechanoreceptors, which are going to detect that stretch and distension. And then that sends signals up to the CNS, which communicates back with the GIT via autonomic nerves. And these can be either parasympathetic fibers or sympathetic fibers. And parasympathetic, remember, is like your rest and digest. So that's going to increase gut motility and secretions and relax smooth muscle sphincters so that the food can pass through. That acts via acetylcholine as the main neurotransmitter on muscarinic receptors, compared to sympathetic fibers, which are going to be your fight or flight response. So that's going to decrease gut motility and secretions and constrict smooth muscle sphincters to slow the passage of stuff through the GIT. And that's via noradrenaline on adrenergic receptors. As sort of like a little extra note, the autonomic neuromuscular junction is a little bit special, and that's because the nerves there, they terminate as these varicosities. So you can see these sort of like bulbous bits to the neuron, and these sort of bulbs or varicosities contain synaptic vesicles with the neurotransmitter, so that as the signal comes down, you get like bulk release through each of these varicosities. And then they act on like clusters of muscular cells. And these muscular cells are electrically coupled. So that means if you get an action potential at one, it will spread throughout a whole bunch of the muscle. And that way you get the coordination that we need for peristalsis. So autonomic innervation stuff. So as general principles, sympathetic, we talked about the neurotransmitter and the function. Parasympathetic, their neurotransmitter and function. The origin of sympathetic fibers, so their presynaptic cell bodies, are going to be in T1 to L2 or L3, depending on the source. Whereas parasympathetic fibers originate from vagus, which is cranial nerve 10, or S2 to S4. More on that in a second. As a general principle, sympathetic nerves synapse at the sympathetic trunk or at prevertebral ganglia. So that's like your celiac ganglion, the superior mesenteric ganglion, inferior mesenteric ganglion, all that. Whereas parasympathetic nerves, they sort of come straight out and synapse near the end organ. So it's presynaptic pretty much all the way down to like the end organ. So here we have a little summary of your autonomic innervation for the gastrointestinal tract. So we've got vagus and pelvic splanchnics as our sort of parasympathetic supply. And the pelvic splanchnics originate from S2 to S4, whereas vagus is your cranial nerve 10. These guys, they synapse at the inferior hypogastric plexus and they supply the hindgut, whereas the vagus does parasympathetic supply of foregut and midgut, everything up to the splenic flexure. So you might hear that mentioned in anatomy as something that's like clinically relevant, because the splenic flexure, so sort of the join between the transverse and descending colon, is like the transition point from vagal innervation to pelvic splanchnic innervation for parasympathetics. For sympathetics, there's a little bit more to it. So you've got your thoracic splanchnics, greater, lesser, and least, your lumbar splanchnics, and sacral splanchnics. So we talked about the roots on Friday, and then the greater splanchnic goes through the celiac ganglion, the lesser through the superior mesenteric, and the lumbar through the inferior mesenteric. And it's helpful to know where the ganglion are, because from there, remember that they sort of wrap around the vasculature and follow the vessels to their end organs. So it makes sense that if the greater splanchnic is going through the celiac ganglion, it's going to supply sort of what the celiac trunk supplies, so the foregut. Whereas your lesser splanchnic going through the superior mesenteric ganglion is going to follow like the SMA sort of distribution, so midgut. And then lumbar splanchnics from L1 to L2 follow the inferior mesenteric artery at the inferior mesenteric ganglion to do like hindgut, and then also some like pelvic viscera. The least splanchnic is kind of not really that important. It goes through the renal ganglion, does a little bit of kidney and ovary supply, but like overall, we don't really care as much about that one. Sacral splanchnics, so we said that these are still sympathetic fibers, so they're still going to originate from the thoracolumbar sort of area, so within these sort of nerve roots, but they'll exit a little bit lower down, and they go via the inferior hypogastric plexus, mixing with these pelvic splanchnic fibers to supply sort of the same type of stuff. Here are some nice pictures that I just threw in to see if they help you. They provide a really nice summary. So this here on the side, you've got the red being like your sympathetic supply. And this really shows everything from like all the way up from the eye down to like the rectum. But really, we're just sort of looking at this gastrointestinal area here. So you can see your celiac ganglion, superior mesenteric ganglion, and inferior mesenteric ganglion, and then each of their supplies, and also the nerve roots that are going into each. Whereas on this side with the blue, you've got parasympathetic. Again, a lot of stuff up in the head and neck that we're not so concerned about at the moment. But the main one you have to worry about.
Sorry, am I frozen? And you like a little bit.
Oh yeah, it's
First,
my
like
internet. Where did I freeze from?
Just after inferior mesenteric ganglion, I think.
Like on the previous slide or on this slide?
No, on this slide.
Oh, okay. I was like, wow, that was really long. Yeah, all good. So inferior mesenteric ganglion here, and then you've got the nerve roots showing as well. And then on the blue side, you've got parasympathetic fibers with a lot of like head and neck stuff that we're not so worried about. And then the main one that we care about is cranial nerve 10, so vagus nerve. And that comes down to supply a lot of stuff in the thorax, but also our GIT. And this is all parasympathetic supply. And then you also have like sacral down here. So this was S2 to S4 sort of origin. And that's doing like your hindgut up to the splenic flexure here in the large colon. Cool. So we have innervation from the CNS, but we also have local enteric innervation of the GIT. So when your sensory neurons detect the distension in the gastrointestinal tract, they project up to the CNS, but they also project into the muscular wall to the myenteric plexus and the submucosal plexus. So the naming of these is really helpful to think about like the function of them. So we know that myo means like muscle. So it makes sense that the myenteric plexus is going to supply the muscle. So it's located in between the circular and longitudinal smooth muscle, and it controls motility through the gut. And so it makes sense that that would run along the entire length of the gut because we have muscle the entire length of the gut. Whereas the submucosal plexus is going to be located in the submucosal connective tissue, and mucus is like a glandular secretion. So submucosal would be controlling secretion from glands. And that's going to help with absorption of nutrients and dilation and all that sort of stuff. And these are only located in discrete regions of the gut, basically areas where it's important to control secretion from glands. As a general principle, excitatory neurotransmitter would be your acetylcholine, and the inhibitory neurotransmitter would be nitric oxide. Okay, so here's like a little diagram. Sorry, it's really blurry, but we have our lumen here and the bolus, and then that's going to be detected by sensory neurons here, specifically mechanoreceptors, which will project up to the CNS, and also into the submucosal connective tissue and the muscularis layer. So here we have the myenteric plexus, which would be in between our circular and longitudinal muscle. And here we have the submucosal plexus. And these are going to control like your glandular secretions. Okay, so we've talked about peristalsis and motility through the gut, but we also have to be aware of the sphincters, which are going to basically cut off sections of the GIT to prevent the food from refluxing back up. So these are a mixture of smooth and skeletal muscle. So most of them are smooth muscle sphincters, so that would be autonomic control, versus just the upper esophageal and the external anal, which are skeletal muscle sphincters, so under somatic control. And I mean, the naming of these, it kind of makes sense as to where they would go. So you've got the upper esophageal, which is like up here when you're swallowing, lower esophageal at the lower end. And this one's really important because this is what's implicated in GORD. So reflux, you get weakening of the lower esophageal sphincter and reflux of the gastric contents up into the esophagus. At the end of the stomach, we have the pyloric sphincter, which makes sense because you have the pylorus of the stomach here. And then the hepatopancreatic or sphincter of Oddi is the one that's at the junction of the common bile duct and the duodenum. So that's what's controlling biliary flow into the duodenum. Then we go all the way down to the ileocecal junction, where you have another sphincter, and then all the way around to the internal anal and external anal. As a general principle, sympathetic activation, remember, is going to try to reduce sort of movement through the gut. So that would contract these sphincters versus parasympathetic activation, we're increasing movement through the gut. So that would be relaxing the sphincters. And the myenteric plexus also can relax the sphincters through nitric oxide. Remember, that's your sort of inhibitory signal. So summary, we've got sympathetic nerves and parasympathetic nerves from the CNS, myenteric plexus and submucosal plexus locally. Motility, secretions, and sphincters. You can have a look at this in your own time, but it sort of summarizes the impact of all of the nerve types on sort of the functions of the gut. Okay, so then we look specifically at, oh yeah?
Do learn that, because it's kind of important, or slide before.
Here?
Yeah, and Greg, remember the idea that sorry.
I can't really hear
Just
you.
like the table slide. Ignore me then. Don't.
Can you guys hear him, or is it just my internet?
No, I think it's Kevin's internet.
Oh, okay.
I'm not sure if you mentioned, but myenteric is motility because M&M and secretions for submucosal. So that's like an easy way to remember that.
Oh, that's good. Okay, we'll keep going then. Yep, so motility in the small intestine. You get two types of like contractions, I guess. And one of them is the segmenting contractions, and their primary role is to sort of mix the food back and forth. And then you're going to get migrating myoelectric complexes, and their primary function is to push the food along into the large intestine. So your segmenting contractions, they get triggered once stretch receptors in the small intestine sort of detect the presence of food. And these, they sort of wriggle the food back and forth. It's like 3 steps forward, 2 steps back sort of idea. And that's going to provide enough time to facilitate all of the chemical digestion and nutrient absorption that has to occur here. Once you've gotten all that nutrient absorption and the chyme pH is sort of gradually increasing, it transitions to migrating myoelectric complexes. And these are like big peristaltic movements where each wave sort of pushes the food 60 centimeters down along the small intestine. And so that's going to try to clear out the small intestine, and then they stop once you get a new meal in the stomach so that the segmenting contractions can pick up again. In the large intestine, you have a very similar principle. They just have different names. So instead of the segmenting contractions, we have haustral contractions. So the haustra are these like sacs that give like the sort of bulbous segmented appearance to the colon. And when you get chyme entering the haustrum, you get that distension picked up by the stretch receptors and activation of the myenteric nerves. And this creates a mixing type contraction again. But here the main function is to assist with water absorption, because that's really the primary function of the large intestine here. Then when we're ready to sort of like defecate, you get mass movements. And this is again like a sort of coordinated peristaltic contraction of larger segments of colon. They last about 30 seconds. You get brief loss of the haustra during the contraction. And these occur 3 to 4 times a day, up to 10 times a day. And that's going to push the food from the cecum all the way around to the sigmoid colon and into the rectum. Now, once the food reaches the rectum, you don't necessarily always want to defecate, but the internal anal sphincter, which is under autonomic control, responds just to the presence of the feces. So once you get mass movement, pushing the feces into the sigmoid colon and rectum, you get the stretch receptors detecting distension again, and they're going to activate parasympathetic nerves, and you get ultimately relaxation of the internal anal sphincter. You also get some increased mucus secretion, so that would be via your submucosal plexus, in order to sort of coat the stools and help facilitate passage. But obviously we don't want to defecate every time a mass movement happens, and so that's why it's important that we have somatic control of the external anal sphincter. And so this only responds to signals from the cerebral cortex once you consciously decide that you're ready to go. And then that cerebral cortex signal allows these motor nerves, these muscles here to sort of relax, and then you get opening of the external anal sphincter. If you have passage, like a mass movement, passage of the feces into the rectum, and the internal anal sphincter has sort of relaxed, but the external anal sphincter remains closed, after some time, the feces sort of like retreats back into the sigmoid colon, and that's called retrograde peristalsis. And it will just wait until the next mass movement comes along and sort of tries again. Okay, so now we get into secretions. So we're going to start from saliva up the top and then work our way down to like pancreas. So starting with saliva, here are like some facts about it. I don't think you have to worry too much about knowing this sort of stuff, but the idea is just that like it's a relatively alkaline sort of secretion, and they come from salivary glands, and you have 3 extrinsic pairs. And so these are sitting outside of the mouth. You've got the big parotid gland here, and then you've got submandibular under the mandible and sublingual under the tongue, as the name sort of suggests. And these have little ducts that project into the mouth to carry the saliva in. And then you also have intrinsic buccal glands, but we don't worry so much about those. Much of saliva is water, but it does also contain calcium phosphate, which strengthen your teeth, as a fun fact. And then more importantly, you've got like salivary amylase, which begins carbohydrate digestion. We have a question. Can different parts of the small intestine be undergoing different types of contractions at the same time? As far as I know, no. It's sort of like a coordinated movement for the entire small intestine. But correct me if I'm wrong. That would be my understanding.
That's kind of how I think of it as well. It's kind of like it does that, go through, and then it kind of like mixes and then goes through and then mixes and then goes through. Yeah,
because remember you've got that like electric coupling, the sort of syncytium, where like if you have an action potential in one area, it sets off action potentials in the surrounding cells. So it's always going to be like a coordinated sort of movement. You don't have independent innervation of like bits of the small intestine, if that makes sense. Okay, control of salivation. So parasympathetic nervous system, is it coordinated between the small intestine and the large intestine at all? Sort of. So the idea is that like we get the, if I go back, we get the migrating myoelectric complexes that are going to push the food into the large intestine. And so as soon as you get this, you're going to therefore get haustral contractions, right? Because that's going to then stimulate the haustral contractions. So there is a little bit of relationship just in terms of like the passage of the food, but innervation wise, they are still independently innervated, if that makes sense. If I fail to answer the question well, just ask it again or ask it differently and I'll hopefully try again. Okay, so back to salivation. The parasympathetic nervous system is primarily responsible for promoting salivation, and it's triggered by all sorts of stuff, mainly sort of like preempting food entering the gastrointestinal tract. And then we also have the sympathetic nervous system that does sort of promote a little bit of saliva output, but it contributes much less than the parasympathetic nervous system. So it's predominantly a parasympathetic control. And so from there, you can kind of assume what you're going to be doing in order to treat salivation problems. So if we have excessive salivation or drooling, you can use a muscarinic receptor antagonist to block the parasympathetic supply. Versus if you have insufficient salivation, xerostomia, you would use a muscarinic agonist to increase parasympathetic supply.
Elena, question.
Oh, yeah. So if you
Yeah.
eat another meal, segmenting contractions will commence again, and if there's still food lower down, it won't get moved through quickly. So if you eat another meal, segmenting contractions. Yes. So once food enters the small intestine, the segmenting contractions will begin. So I guess if you're sort of mid MMC and like there's still sort of food sitting in the distal portion of the small intestine, you just sort of get the segmenting again, and then eventually, once that's been digested, coordinated movement, and that sort of like pushes them both through, if that makes sense.
Yeah, the general idea is like you're not gorging yourself, I guess, in the normal phys. It's like you eat something and that goes through, and then you eat something again, like a next meal. Like once it clears through, that's like theoretically when you eat again. But if you're eating quite quickly after you've just eaten, then they can kind of mix together as the same meal sort of thing.
Yeah, I think also to note is that it's not going to send it all the way through your intestines in one movement. So you're going to get quite a few of each one in succession, is what I've understood. Yeah, so it might move
so like each,
and then it'll go segmenting again, and then it might move it a little bit again.
Yeah. So like each migrating myoelectric complex might not necessarily get food from like the duodenum all the way down to the cecum, right? So you might have like a couple cycles of segmenting contractions and migrating myoelectric complexes before it makes it all the way through. All right, so now we get into gastric secretions. So that was saliva. And with gastric secretions, 1.5 liters per day, the important thing to know is that it's very acidic. And they come from gastric glands, which are located in gastric pits, which are in like the epithelium, so the wall of your stomach. And so these gastric pits, they're lined by a variety of different types of cells, and each one has its own role in terms of secretion. So mucus cells, they're going to secrete mucus, and that's really important, and I'll get to that in a second. Parietal cells, they secrete your hydrochloric acid and intrinsic factor, both also really, really important. So this is producing that acidic pH that's so sort of characteristic of gastric secretions. Intrinsic factor is essential for absorption of vitamin B12. So some people might have like autoimmune destruction of parietal cells, and that means that they'll lack intrinsic factor, and then they get problems with absorption of B12. Chief cells, so like these guys here, pepsinogen, and pepsinogen is the inactive form of pepsin, which is an enzyme that breaks down proteins. So these are important for helping you break down your proteins. And then we have enteroendocrine cells. So these sort of act locally within the gastric pit on other cells within the gastric pit. So we have enterochromaffin-like cells, ECL cells, that secrete histamine, and G cells that secrete gastrin. And both histamine and gastrin then act on parietal cells to sort of stimulate acid production. The acidity of gastric juices is really important for a couple reasons. One of them is that pepsinogen, which is our proteolytic enzyme, is only activated at low pHs. So we need it to be a pH of like around about 2, but definitely below like 4 or 5 in order to actually get activation of that enzyme. The enzyme also, the acidity also helps kill ingested bacteria and break down cellulose. Okay, so a little bit of sort of biochem stuff on how we actually get acid secretion at the parietal cells. So if you picture that this here is a parietal cell, don't worry about the numbering's a bit arbitrary to be honest, but if we picture that we have a parietal cell here, the lumen of the stomach, and then we have like the bloodstream here. So this is like the basal membrane of the parietal cell. Within the parietal cell, we've got water and carbon dioxide from our cellular respiration, and these are going to combine to form carbonic acid, H2CO3, and that dissociates into bicarb, which is basic, and protons, which are our acid, right? This bicarb gets exchanged at the basolateral membrane with sort of this chlorine bicarb exchanger. So the blood here becomes more basic as it passes through sort of the stomach supply. And that's because it's increasingly getting bicarb from the parietal cells. In exchange for the bicarb, the parietal cell takes in chlorine, and that chlorine gets sort of passively diffused back out of the lumen of the stomach, just down its concentration gradient. We also have potassium in the cell, which sort of passively diffuses outwards as well, and then it gets actively brought back in exchange for protons at this hydrogen potassium ATPase pump, and that is what we call our proton pump. So it's exchanging protons for potassium, and that potassium just flows back out passively again to sort of create this loop. So then once you've got your protons and your chlorine in the lumen of the stomach, they join together to form hydrochloric acid. Okay, so we've talked about how we get the acid production in the parietal cell, but we also need to know like how to control the amount of acid that is secreted, because at rest when you're not eating, you sort of want less acid to be sitting in the stomach, and then once food comes into the stomach, you want to be releasing more acid in order to sort of compensate for that. So there's a couple different mechanisms that act on parietal cells to increase or inhibit acid secretion. So we have gastrin from our G cells, we have histamine from our ECL cells, and we also have acetylcholine, and that's from either the myenteric nerves or the vagal parasympathetic nerves. So parietal cells can be stimulated by those local sort of endocrine cells and also from the CNS and local enteric innervation. Each of those three will act to increase proton pump action and therefore acid secretion. We do also have an inhibitory signal from somatostatin. But as sort of like a side note in terms of clinical applicability, when we're trying to decrease gastric acid secretion, it wouldn't be very much use to just try to target gastrin or just target histamine. So antihistamines, they can theoretically decrease gastric acid secretion because you're cutting off this stimulation here, but you're still going to get signals from the gastrin and the acetylcholine. So it's nowhere near as effective as just cutting off the action over here. So that's why we use PPIs, proton pump inhibitors, to directly cut it off here, and then it doesn't really matter what kind of signals you have coming into the parietal cell. Okay, phases of gastric acid secretion. So we talked about control and the kinds of things that impact gastric acid secretion, but then what impacts these things? So we have the cephalic phase, which is, cephalic means like brain, right? So this is stimulated by like the brain and our recognition of food and our like anticipation of food. And so once you get your sort of 5 senses anticipating the food, we get activation of the enteric and the parasympathetic systems, and that acts on your parietal and G cells. So the increased ACH and the gastrin from the G cells are going to stimulate your parietal cells, so ACH and gastrin, and then that is going to sort of then act on the ECL cells again to secrete histamine and further increase acid secretion. So it's kind of like a positive feedback sort of mechanism where the parietal cell stimulates its own stimulation. Then you have the gastric phase. So this is local stomach activation. So it's activated by distension of the stomach, the increased pH, and the presence of partially digested proteins. And that's again sort of going to do a similar thing to the cephalic phase, and you're going to increase your acid secretion via all of those different stimulating signals. The intestinal phase is your inhibitory mechanism, and it makes sense because once the food has passed out of the stomach into the intestines, you don't really want to be overproducing acid in the stomach anymore. You want to sort of calm down the stomach. So once you get distension of the duodenal wall and decrease in pH, so the acidic chyme is coming into the duodenum, you get intestinal endocrine secretions, which we don't worry too much about, but ultimately you get these guys and inhibition of the vagus nerve, and that is all going to decrease gastric acid secretions. So the cephalic phase stimulated by like the brain, the gastric phase stimulated by the stomach, and these are both sort of increasing gastric acid secretion. The intestinal phase stimulated by food in the intestine, and that's going to decrease your gastric acid secretion. Okay, the gastric mucosal barrier. So we mentioned before the mucus cells, which are producing mucus to sort of line the stomach. And that's super important because it's one of the main protective mechanisms. The pH of gastric juices is very, very acidic. And so if that is just sort of in contact with exposed tissue, it will erode the tissue. So we have this bicarb-rich mucus secreted by goblet cells in those gastric pits in order to protect our stomach lining. There are a couple other protective mechanisms. You've got tight junctions between the epithelial cells to try to decrease like seeping of the acid into deeper layers. And you also have like undifferentiated stem cells that are very actively and rapidly dividing to replenish any sort of damaged epithelium. Ultimately though, if you have any failure of these protective barriers, mainly the mucus really, you get ulceration. So this here would be like a stomach ulcer. And that's like a very painful sort of sensation in the stomach, and it's caused by the acid eroding into the tissue. If it's really, really bad, it can erode all the way through the tissue and perforate. And then you get spilling of the gastric contents into the abdomen, which is very, very bad. And gastric ulcers can be caused by a whole bunch of different things, but most commonly by H. pylori infection. Other gastric acid problems. So remember, I mentioned before, GORD, so gastroesophageal reflux disease, is mainly an issue with failure of the lower esophageal sphincter. And we basically treat it with PPIs to decrease gastric acid secretion. You can also get problems with defective secretion. So people who've had gastrectomies, so surgical removal of the stomach, they often have some issues with secretory capacity and therefore absorptive capacity. So same with autoimmune gastritis here. This just means you're getting autoimmune destruction of the stomach cells. And the main mechanism that you need to know is intrinsic factor deficiency. So remember, intrinsic factor is secreted by parietal cells. And so if you have autoimmune destruction of parietal cells, or you've surgically removed a bunch of stomachs, so there's just less parietal cells, you're going to get less intrinsic factor. And intrinsic factor is very important for vitamin B12 absorption. So you get vitamin B12 deficiency. Vitamin B12 is important for red blood cell formation, and so therefore you get pernicious anemia, macrocytic anemia specifically, and also neurological disturbances, and this can be a presentation of vitamin B12 deficiency. Okay, so now let's move on to bile. So bile is a pretty alkaline, so slightly alkaline substance. It's continuously secreted by the liver into the biliary system, and it's stored in the gallbladder. And then whenever you get a meal coming into the duodenum, you get this sort of big contraction of the gallbladder to push a whole bunch of bile in. Bile contains a whole bunch of different stuff, bile salts, and these are important for helping to emulsify fats. And then you've got a couple other things that help with the fat emulsification, electrolytes, specifically bicarb, to neutralize the acidity, and bile pigments, bilirubin. These functionally don't have that much importance, but this is an important sort of excretory mechanism for heme. Functions of bile, so we're neutralizing the acidic juices with that bicarb, emulsifying the fats with the salts and the lecithin, and also this is helping to excrete certain liver products, the bilirubin and the cholesterol. So a little more biochem. This is honestly not that important to know, but it was taught to us in a lecture, so I thought I'd summarize it here anyway. But we talk about sort of like the bile salts that are present in the bile, and then there's sort of various other terms for the different stages of the production of bile salts. So it all starts with cholesterol in the liver, and this is important because bile is sort of like our only real way of excreting cholesterol. So if you have like hypercholesterolemia, you can sort of attack that or try to change that by altering this pathway. Or you could have hypercholesterolemia due to a defect in this pathway. So cholesterol in the liver is processed into primary bile acids, and then these are conjugated to become bile salts. And then the bile salts are what are secreted into the GIT with your bile here. So bile salts are coming into this system. Once in the GIT, anaerobic bacteria unconjugate the bile salt, and then it becomes secondary bile acids. And then these guys can then get absorbed later on in the GIT. So I think I mentioned that later on. We also have regulation of bile synthesis. If you take one thing away from this slide, this is probably the line to take. And that is that the rate limiting step of bile acid synthesis is the conversion of cholesterol into 7-alpha hydroxycholesterol. So that first initial step by 7-alpha hydroxylase. So problems with 7-alpha hydroxylase are going to affect your bile production and bile secretion. All right, So this is sort of important to understand the function of the bile. So we have like a big dietary fat globule here sitting in the intestines. The bile salts and the phospholipids are also hanging out in the intestines, having been secreted via the sphincter body. And then we have those segmenting contractions within the small intestine, which are mixing up the fat globules. And in doing so, it sort of momentarily breaks up the droplets, right? It's like that idea of like, if you have oil and water, they don't mix. But if you like aggressively shake the mixture together, they momentarily form droplets before coalescing back together. So bile salts, their function is that while you get those momentary droplet formations from the mixing, they sort of quickly surround the emulsion droplets to prevent them from recoalescing into a big globule. So the bile salts and the phospholipids surround our emulsion droplets to keep them small. And then we have our emulsion droplet over here, and it's full of triglycerides from our diet. And the presence of these bile salts and the lecithin gives more surface area for lipase to get in, right, compared to like a big fat globule. So the lipase is going to be able to get into those triglycerides and break them into monoglycerides and fatty acids. And these can either diffuse into our intestinal epithelial cells to be absorbed, or some of them form like micelles, which are basically like mini versions of the emulsion droplets. And these guys aren't absorbed, but they sort of go between being micelles and free fatty acids and monoglycerides according to like an equilibrium type thing. Okay, so that is all of that basically just written out in words if you want to read it while looking at the picture. So bile secretion and recycling. So I guess we have to talk about the stimulation of bile secretion. So we mentioned it's sort of stored in the gallbladder here, continuously produced by the liver, but how do we get the signal in order to make the gallbladder push it into the duodenum? And so that comes from the presence of chyme in the duodenum. So chyme being that sort of food soup that you get out of the stomach once it's sort of been slightly broken down. And it's very acidic because it's just come out of the stomach, and it's also high in fats and also other nutrients, but fats are really what we care about here. So that acidity gets detected and causes secretin secretion, and the fat gets detected to cause cholecystokinin secretion. And these hormones have important functions. So they enter the bloodstream and they flow all the way around the body, but secretin acts on our liver to increase bile production, whereas cholecystokinin acts on the gallbladder, and that's what's going to cause contraction and also relaxation of this sphincter down here to allow the bile salts to pass through. CCK also has a whole bunch of other functions. It acts on the pancreas to increase like the activation of pancreatic lipase. It slows down the stomach churning. It constricts that pyloric sphincter to decrease movement into the duodenum. And so it has sort of a lot of functions in the GIT. But in terms of bile secretion, it's really important for causing contraction of the gallbladder. Now, once your bile salts are in the duodenum, the vast majority of them actually get reabsorbed. So you get that unconjugation by the anaerobic bacteria, blah, blah, blah. And 95% of the bile salts actually get reabsorbed back into the blood, pass through the hepatic portal circulation and back into the liver. So they get recycled and then reused. But some of it, 5%, gets broken down by bacteria and excreted in the feces. Okay, so what actually makes up bile? So we mentioned the bile pigments or the bilirubin that is present in our bile. And I mentioned that that's sort of like a product of hemoglobin breakdown, and that's an important excretion pathway for the hemoglobin breakdown. But you do have to know some of the steps in between. What happens with the secretion of bile if the gallbladder is removed? Yeah, good question. So we said that liver constantly produces bile versus the gallbladder, which is just sort of a little storage pouch. And then you get contractions so that you get like a mass amount of bile. If you've had the gallbladder removed, it just means you don't have that storage capacity. So you don't have the ability to like suddenly influx a big bulk of bile. But you still have the liver, which is constantly producing it. And we still have secretin, which is going to increase bile secretion by the liver. So you still get some bile and you still therefore get some fat absorption, but you just get less. So people who've had their gallbladders removed, cholecystectomy, they have some decreased fat absorption capacities. So they get like steatorrhea, which is like your greasy, fatty stools, and they might have some issues with fat-soluble vitamins and deficiencies there because they're just less able to break down the fats and to sort of absorb the fats there. But they still do have some function because the liver is still there and it's still producing the bile.
Yeah. I think clinically what we got told last year was that they have a slight change in their diet, so they can have to reduce their fat intake and all that kind of stuff, and don't eat macros probably. But overall, their quality of life is quite similar. So yeah, like Alina said, the gallbladder is just there to give you an extra help, but it's not critical, I guess.
So it's in avoiding high density fat meals, but you can eat a low level of fats pretty much.
Yeah, so they can still cope with a small amount of fat, but to reduce those adverse effects, the steatorrhea, they get told to do a low fat diet. All right. So back to the bilirubin. So this pathway here, so we start off with hemoglobin, which is in all of your red blood cells. And when the red blood cell dies or ruptures, you get release of this hemoglobin into the bloodstream. And the hemoglobin gets broken down into heme and globin, conveniently named. And the heme is oxidized into biliverdin. And biliverdin is a greenish pigment. So that's why bile is kind of greenish. So some of that biliverdin, it's unconjugated, it's not very soluble, and so it has to be bound to albumin in order to sort of flow around the blood. So that's why we have protein bound unconjugated bilirubin in the plasma. Once it gets around to the liver will conjugate the bilirubin, which makes it a little more soluble, and then you get your whole release into the biliary system and into the small intestine. And then we mentioned 95% of it actually gets reabsorbed, but 5% is broken down and converted into urobilinogen by the bacterial flora in the intestine. And that gets excreted as feces, which gives feces its darkish color. And then some of it in the general circulation, if it's been reabsorbed, it gets excreted by the kidneys in the urine, and that would give urine its darkish color. So if you have a blockage of your bile duct here and you're getting obstructive jaundice, what happens is we get less bilirubin moving into the into the intestines, and therefore less of that urobilinogen in the feces, and you get pale stools as a sign of obstructive jaundice. You also get dark urine, and that's because if you've got blockage here, then you're actually going to have more of the bile floating around in the bloodstream, and so more of it is actually going to be excreted by the kidneys and in the urine. So we get darkening of the urine and pale stools in obstructive jaundice. Okay, pancreatic secretions. I think this is the last one. But the pancreas, Kevin did mention on Friday, has a couple different functions, endocrine and exocrine. So endocrine being that hormones which act inside the body, so your insulin and your glucagon, and that's for controlling blood glucose. And then exocrine being secretions that go outside of the body. So even though the gastrointestinal tract is kind of inside our body, it's really considered an external environment because it's freely in contact with the external world via the mouth and the anus. So these are exocrine secretions, and your exocrine tissues are sort of in this kind of configuration. We've got acinar cells here, which are kind of the bulbous endings, and then we have ducts, and the ducts themselves are lined with cells that also have secretory functions. So the acinar cells here, they produce your pancreatic enzymes, so trypsin, chymotrypsin, carboxypeptidase, all of that stuff. And the main thing to note is that its inactive precursors are secreted, and then those inactive precursors are activated once they get into the duodenum. And that's to prevent those enzymes self-digesting the pancreas. Because if it's a proteolytic enzyme, it breaks down proteins, then if they're active inside the pancreas, they're going to break down the structure of the pancreas. And so you can get clinical pathology here where you get obstruction of these ducts and therefore sort of stasis of all of these enzymes within the pancreas. And the high pressure can basically trigger the enzymes to activate, and then you get autodigestion of the pancreas, which causes pancreatitis. But in normal physiology, the inactive forms are secreted. And remember, your pancreatic duct joins with the common bile duct, the common hepatic duct, to make your common bile duct before it joins into the duodenum. And then we also have the ductal cells. So the acinar secretions come through the ducts, but the ductal cells themselves have a function, and that is to produce bicarb. And so that's why your pancreatic secretions are going to be alkaline and also full of those digestive enzymes. Okay, regulation of pancreatic secretion. So regulation of the pancreas is by the same hormones as regulation of a bile secretion, but they just have slightly different functions here. So we get the same sort of mechanism with chyme emptying into the duodenum from the stomach. The acidity triggers secretin secretion, fat triggers cholecystokinin secretion. But here, secretin causes the pancreatic and bile ductal cells to secrete their bicarb-rich solution. And then the CCK stimulates the acinar cells to secrete the pancreatic enzymes. And remember, also relaxation of the sphincter body. So ultimately, what you're getting with the CCK and the secretin is movement of that bicarb, the pancreatic enzymes, and the bile through the relaxed sphincter body into the duodenum. You also get a little bit of vagal stimulation, and that's going to help to like increase pancreatic enzyme secretion. But mainly we talk about these local hormones, the secretin and the cholecystokinin. Okay, so that's GI physiology. Any questions about that? If not, we'll move on to micro.
Alrighty. I will share my screen.
Yep, I'll stop sharing. Should we change recording or keep the recording?
I think we'll keep it going on
You
yours.
can keep it. Just easier.
Sure.
Can you guys see that? Ooh.
Yeah, I can see.
We're good?
Yep.
All right. So I'll cover GI micro first and then pharm after. But basically, I guess the structure of the presentation will be going through vomiting, diarrhea, and acid reflux and causes, as well as treatment with or pharmacological treatment. In terms of micro, a lot of the stuff is pretty much just buzzwords, so we won't get bogged up in all the details, I think. But I'll try and give you some principles to keep in mind, as well as the important things to know about each of the, I guess, microorganisms that we're going to be going through. So we'll start with diarrhea. This is just a brief explanation of what diarrhea is, but the clinical definition is 3 or more loose stools in 24 hours. But we've also got this other sort of definitions that people do use from time to time. So they say either excess stools in terms of mass, or more frequent stools, or stools that just assume the shape of their container. So diarrhea kind of fits into all those different categories, and it's hard to define it that way. We can either define it as acute, persistent, or chronic. So acute is 3 to 7 days. Chronic is more than a month, and persistent is sort of in between that. There's sort of this 3-day rule that people use for diarrhea. So if you've got diarrhea for less than 3 days, it's generally community acquired. So it's a bit more self-resolving. But if it's more than 3 days, it's more likely to be hospital acquired. It's more likely to be a bit more resistant, or a C. diff infection because it's hospital acquired. The types of diarrhea, so we can get blood in the diarrhea, we can have mucus in the diarrhea, we can have watery diarrheas as well, but that's a bit more specific. We don't see that as often. Or greasy diarrhea, so more fatty stools. And things that you need to be asking when you're in diarrhea, so you ask your GIT systems review, which JJ will talk about later, I'm sure, if they've been traveling. So that's in terms of gastro or salmonella or other sort of more exotic pathogens, whether they've been exposed to other people with diarrhea, or infected from other people, their immune status as always, and whether they've been taking drugs which would make them more susceptible to getting diarrhea. In terms of investigations, there's a bunch of different investigations, but the main ones you do are, so there's stool samples, so you do your traditional microscopy, culture, and sensitivity. So trying to determine what organism it is, and what you might want to treat it with based on its sensitivity. There's these specific selective cultures, which are hard to remember, but McConkey is selective agar, selenite is for salmonella in particular, so S and S, XLD, so you see sort of pink pathogenic growth with black centers on them. So the way I remember it is like Blackpink, if people watch Blackpink or listen to Blackpink or whatever. You've got ova, cysts, and parasites, which are much more uncommon, and they're not traditionally done as like first line. Or you've got PCR of your stool. So that's probably one of the best ways you can determine in like causes of diarrhea. It's quick, it's sensitive, You can detect bacteria as well as viruses. But the main problem with that is you can't do your antibiotic sensitivity test because you haven't got a culture growth of bacteria on agar to test them on. So that's the main problem there. And that's why we still sort of do our microscopy culture and sensitivity still. And you've got these other ones, so toxin assays specifically for Clostridium, serology, and blood cultures, if you want to check for like typhoid or sepsis and things like that. Treatment is often self-resolving. Sometimes you might need antibiotics if it's more hospital-acquired, but generally not. You just need sort of fluid management, fluid replacements. Okay, so in terms of telling the difference between small bowel and large bowel diarrhea, there's a few different sort of key elements you can look at. So small bowel diarrhea is more voluminous, whereas large bowel diarrhea is more sort of frequent small volume stools, but you still get excess in both. Large bowel diarrhea is more bloody, whereas small bowel diarrhea is more watery. Small bowel is more smelly, and it's more prominent in vomiting. It's more likely to go up the other way than coming down through the fecal route, and dehydration is more common in that sense. If you have more vomiting, you're more likely to get more dehydrated. Causes of diarrhea. So this isn't that important. I wouldn't remember any of this, but for those who are out there and budding microbiologists, like there you go. But we won't spend too much time on this. All right, so norovirus. This is probably one of the big ones to know. Cause of gastroenteritis, a big cause of gastroenteritis, and actually diarrhea. It's most common in adults, and it's pretty much it's very infective. Mainly, you see it in hospital wards. So when there's those outbreaks of gastroenteritis in the hospital wards, it's most likely to be norovirus. And you don't generally need to treat it. You just sort of need to isolate yourself, prevent infection towards other people. Or if you want, you can just have fluid replacement. So taking oral rehydration therapy, so like hydrolyte, things like that, drinking lots of water. And then there's rotavirus. So this is more common in children. And it's the commonest cause of non-bloody diarrhea, actually. But you've got sort of similar things with a fever. You've got diarrhea, vomiting. It's less severe than norovirus, but it's more common. And it's sort of downtrending in terms of frequency because we have vaccines available now. And again, you treat generally with supportive therapy. Most people don't need antibiotics for that. E. coli is another cause. So there's two types of E. coli. There's enterotoxigenic and there's enterohemorrhagic. As the name suggests, enterohemorrhagic is causing your bloody diarrhea. So you're more likely to bleed from that because it specifically secretes toxins which are sort of lysing your vascular endothelial cells, so that will cause blood to come out of your stools. Whereas the enterotoxigenic one doesn't. It more sort of causes that osmotic shift in the content of the diarrhea, so more water is likely to be secreted in the large bowel, and it's sort of associated more so with travelers' diarrhea. So people go to like foreign countries. I think they call it like Delhi belly or Bali belly. That's what they generally get, and self-limiting. All right, salmonella. So I guess the big thing here is if you see chicken or eggs, if people are eating raw chicken, raw eggs, eggs are more uncommon, but mostly raw chicken, it's commonly going to be salmonella, gram-negative rod-shaped bacteria. I think generally most bacteria in the gastrointestinal tract are going to be gram-negative rod-shaped bacteria. If you had to guess anything, guess that. There's a few that aren't, but
I think, like, generally speaking, they're all gram-negative rod-shaped, like gram-negative bacilli, unless it's enterococci, in which case it's like gram-positive cocci.
Yeah, exactly. I think, and that has implications for how we treat with the antibiotics as well, which we'll talk about later. But yeah, if you see chicken or eggs, it's generally salmonella. That's sort of the buzzword on the exam. And you can see they use it specifically, they test it with those XLD plates. So that black and the pink on hydrogen sulfide. There's another type of salmonella that's more associated with typhoid fever, so salmonella typhi. And this is sort of it's much worse, I guess, because you get that sort of systemic, it's more likely to cause systemic infections. So you get like your classical signs of sepsis, so fever, chills, and then your constipation and diarrhea from the gastrointestinal system. And this is not as self-resolving. So you need to treat with more of those broad spectrum antibiotics, so your ceftriaxone, ciprofloxacin, and your azithromycin. So azithro and cipro are more for gram-negative organisms, and then your ceftriaxone is more covering sort of broader causes if you're not exactly sure that it's Salmonella in particular. Campylobacter, so again, this is another cause of diarrhea, and the main complication to know from this is that it causes Guillain-Barré syndrome. So after people get infected by Campylobacter, there'll be a short period, and then they'll sort of get this ascending bilateral neuropathy. So they'll get weak in their legs, and then they'll get weak in their thighs, and then it'll come up their arms, and then eventually they might get like respiratory distress or something like that. So that's sort of, if you see like, they got infected and then and again, you're treating with those macrolides for gram negatives, fluoroquinolones and gentamicin. Shigella, another gram negative rod one. It causes sort of that painful and severe bloody mucus diarrhea. And it's more prevalent in men who have sex with men populations. I think that's probably the one to note. Clostridium. I just realized that picture is going to be there in the recording. Okay, so toxin A and toxin B. Clostridium produces both of those. And I think the most important thing to notice here is that it's an opportunistic infection. So Clostridium normally sits in our gastrointestinal tract, but if we're taking antibiotics for other infections, so say we have pneumonia or UTIs or something like that, it might kill off some of the other bacteria in our gastrointestinal tract, and that might let this Clostridium grow. And so that's why we call it an opportunistic infection, which classically presents after your antibiotic treatment. So if you want to treat that, people generally just stop the antibiotics. They give metronidazole, because Clostridium in general is anaerobic. So Clostridium tetanus, difficile, and botulinum are all anaerobic bacteria. We give that metronidazole because it targets anaerobic bacteria. And becoming, I guess, growing in novelty is fecal transplants to sort of replace your normal gut microflora and sort of restore the balance, I guess, in terms of treating Clostridium difficile infections. Vibrio cholera, so again, gram-negative, comma-shaped bacilli, not as, still along the same lines of gram-negative rods. The most important thing to note is that they cause rice water stools. So you get that heavy secretion of water. It's not super important to note the mechanism of action here, but it's mainly to do with chloride being pumped out, and so water follows the chloride, and so you get that excessive loss of water. And get a treatment with your gram-negative targeting antibiotics, so azithromycin and ciprofloxacin. Giardia lamblia, so this is not a bacteria, it's a parasite. Anaerobic, causes poor absorption of nutrients, and if we get poor absorption of fats in general, that leads us to getting our greasy and fatty and foul-smelling stools. So people have greasy, foul-smelling stools that are floating in the toilets, more pale. That will generally be because of Giardia lamblia, another cause of chronic diarrhea. And then these are other causes of non-infected diarrhea. So I think the most important ones here to notice are drugs and hypothyroidism are probably the big ones for you in particular, but I doubt they'll ask any questions about this. It's just more for your knowledge. All right, vomiting. So what is vomiting? Forceful evacuation of gastric contents via the mouth. So not through the fecal route, through the oral route. And there's certain bacteria associated with that we'll be talking about. So the main ones are Staph, Bacillus cereus, and Clostridium botulinum. We'll talk about Staph first. So Staph, gram-positive coccus, stains purple. It's mainly caused by food poisoning. So meat and dairy products. So meat, so the classic example is like a barbecue. If you leave a barbecue and you don't store the food properly, and it's just sitting outside, then it can get staph contamination. The mechanism it does that is through an enterotoxin, but it's generally self-limiting. Bacillus cereus, so this is a gram-positive bacillus. It's not super important to notice that, but it is different in that aspect. The main way people get Bacillus cereus infection is through rice that's been kept for too long. Basically, you've cooked it, you've kept it in the fridge for maybe a week, maybe 2 weeks. I know it's pretty common for people to do that, but apparently you can get Bacillus cereus infections. I've kept rice in my fridge for a very long time, never got infected, so I don't know how accurate that is, but yeah, that's sort of the main thing to note. And there's 2 types of toxins, and there's toxin 1 that's causing that sort of main problem here. It's the most common sort of issue caused by Bacillus cereus. Clostridium botulinum, so another anaerobic bacteria of the Clostridium family. The main thing to notice here is that it has a different mechanism of action. So it works on our neuromuscular junctions. So it's working on our muscles and how acetylcholine is stimulating muscular contraction at those muscular junctions. So the way it works is it prevents the release of the acetylcholine at those neuromuscular junctions. So because we can't, because acetylcholine isn't traveling across the synapse, it's not stimulating muscle contraction. So we get sort of this paralysis induced with Clostridium botulinum. And vomiting is one of the key presentations there. But you also get this other sort of descending weakness and paralysis associated with Clostridium botulinum. But vomiting can also be a significant part of the presentation. And the main treatment here is antitoxin, to counteract that botulinum toxin. Dysphagia, so common causes of dysphagia. So dysphagia is difficulty swallowing. So if you imagine you have difficulty swallowing, you've probably got either swelling, your esophagus is sort of narrowed, the lumen of the esophagus is narrowed, and that's mainly due to reflux or GORD, causing that esophagitis, or you've got an infection. So common ones in immunosuppressed people are Candida or CMV or HSV. HIV is more so the cause of the immunosuppression, leading towards those infections. Or you can have distressing dysphagia. So people are like crying a lot, or they're in emotional turmoil time of their lives. That can cause dysphagia as well. And the last thing I want to talk about for micro was Helicobacter pylori, because they don't stop talking about that in the exams. It's kind of a common question. It'll be in your SPCs and things like that. But there's just a few things you really need to know about it. It's probably, I think it's the most common cause of gastric and duodenal ulcers. Complications of it are gastric lymphoma, gastric cancer. The diagnosis we use is the urea breath test. It's quite specific for Helicobacter pylori. And the treatment is your triple therapy. So you use a mix of amoxicillin, clarithromycin, and your proton pump inhibitors. And that's mainly, the proton pump inhibitor is mainly for the gastric ulcers you've got, and then the antibiotics towards targeting the Helicobacter pylori. And it's also important to note that it's quite a resistant bacteria, and it's becoming more and more resistant as time has go on, for your own knowledge about the world, I guess. All right, so that's all of GIT micro. Any questions? It's fairly simple stuff, I think.
I think sorry, go ahead.
No. I was just going to say, it's mainly about learning the buzzwords. I don't think it's very high yield to learn all this detail in terms of micro. Like, you'll never really be asked about it. And I don't know if it ever helped you that much in life as a clinician. Sorry, you go.
I think one thing that just like missed my mind last year was that often their morphology is in the name. So if they're asking you, what Staphylococcus looks like. It's a cocci because it's got coccus in there. Same as Enterococci. That's a coccus, so they're the round ones. And same thing with like Bacillus cereus, it's bacillus, and it's literally in the name. So sometimes when you're looking around for clues, you can actually find it.
Yeah, that's a good hint. Like Enterococci also gives you that clue. Yeah. All right.
If it doesn't have cocci in the name, you can generally go by the principle that it's probably a gram-negative rod, because it really is gut bacteria. They tend to be gram-negative rods.
Yeah, exactly. And then there's just the other one to notice that your Clostridiums are anaerobic, and that will get you through most of micro. All right, we'll move on to GIT farm. So GORD is one of the main conditions we talk about. So that's sort of your reflux of stomach acid through your esophagus, and that's what we see, that symptom of heartburn. So sort of the contributing factors of GORD is if your lower esophageal sphincter isn't working. So if you imagine it isn't closing properly, you get that sort of acid is more readily able to come up through the sphincter and into the esophagus. If you've got impaired gastric clearing, it's more likely that acid is sticking into your stomach. So it's more likely that it's going to come up than go forward. Or if you have a hiatus hernia, so that's when your stomach is going through the diaphragmatic hiatus, the esophageal hiatus, I believe. And it sort of just causes that anatomical deformity, leading to more likely gastric acid going back up. And the treatment for GORD is generally, so we always start with lifestyle change, so eat less acidic stuff, changing your diet, drinking less sort of irritants of the stomach, or we can use pharmacological therapy. So reduction of our intragastric acidity, or we increase the mucosal defense. So we protect our mucosa, we protect the stomach layer, or we just reduce the acidity and the offending agents in the stomach. So peptic ulcer disease is the other one that we treat. So this is sort of when we get a breach in the gastric mucosa, ulceration. So we've got damage through the mucosa into sort of the deeper layers of the stomach. And there's sort of three common ones that we need to, three common causes that we need to be aware of. So there's Helicobacter pylori, as we talked about before. There's smoking, which you can't really change unless people stop smoking. And there's NSAID use. And I think I've got a, I don't know if I put a picture. Okay, I've got a picture later on. But we'll talk about why NSAID use is important there. But Alina sort of touched on it, I think, in her talk a little bit. So the main way we treat our peptic ulcer disease is through inhibition of that proton pump that Alina was talking about. So I guess it's more important to look at the picture. So our proton pump is sitting on, so this is the luminal side. This is the basal side. So if we inhibit this proton pump, we're stopping all this gastric acid secretion going into the lumen, the gastric lumen, and we're reducing the acidity of the stomach. And so we're reducing sort of the offending agent, the potency of the gastric acid in causing those duodenal ulcers. And as Alina talked about before, the main principle to note here is that it's more effective to target this proton pump than targeting sort of the stimulating factors, so targeting the gastrin, targeting histamine, or targeting muscarinic antagonist, or targeting prostaglandin. So these are all going to be contributing to gastric acid release. If we target those alone, the other ones are still going to fire off, right? So that's why we target your proton pump inhibitors, because it's more sort of comprehensive in that aspect. Yeah, so this sort of just puts that all into words. So if we stop secretion of H+, we stop secretion of Cl-, and that's from parietal cells, because parietal cells are the ones that are releasing that gastric acid. So the indications for that are generally when people have gastric ulcers, and then your common adverse effects are just nausea, vomiting, and diarrhea, and more rarely people can get gynecomastia or renal impairment. But generally, people are pretty good with it. It's not that, it's generally well tolerated. Yeah. Okay, so if we look at this picture as well, we can see that if we blocked histamine, or if we blocked our muscarinic receptors, or if we stimulated our prostaglandins, we would be protecting our, we would be reducing the acidity in the stomach. So these are other mechanisms we can go about for reducing gastric acid pH. This slide is meant to be here, but this is what we use in Helicobacter pylori. It's a macrolide erythromycin. And the way I remember it, it's similar to erythromycin. So if you want to remember that it's a macrolide, all of these have throw in them. So you throw your Mac into the C because Macs are bad computers. So that's how you remember it's a macrolide. The way I remember that it targets 50S ribosome is based on erythromycin. So Eri sounds like very, and that's very big. So it's not 30S ribosome, it's the 50S ribosome unit because it's a bigger one, it's a bigger number. And we use this in our triple therapy. And again, you get those nausea, vomiting, diarrhea. You can say that for pretty much any antibiotic or any drug as a common side effect. Yeah, okay, so this is talking about, so ranitidine. So this is a histamine receptor. So we're blocking the basal side of the parietal cells in order to stop hydrochloric acid release. So commonly known as Zantac. This is more used in nocturnal acid secretion and duodenal ulcers in particular. Histamine is more prominent at night, and so that's why they use it for nocturnal acid secretion. Yeah, fun fact, there's actually a recent recall of ranitidine because it's contaminated with NDMA, which has long-term risk of cancer. Not MDMA, but NDMA. Yeah, in terms of remembering ranitidine and its mechanism of action, I just remember histamine rhymes with ranitidine, and so that will help you remember it in that aspect. Yeah.
I think it's also kind of interesting to note that your antihistamines for like your hay fever and stuff are H1 receptor antagonists, so they're different receptors that they're looking for. Even though they're both histamine receptor blockers or antagonists, they block a slightly different one, so they do different things as a byproduct.
Yeah, exactly. And we'll talk about that a little later on as well in terms of generations of antihistamines. Okay, so Kralfate. So this is a mucosal protective agent. So instead of reducing our pH in the stomach, reducing the acidity of the gastric secretions and its potency, we're sort of more so protecting the stomach. So the way this works is at an acidic condition, it will sort of form this viscous sticky paste. And that sort of like sits along the stomach, sits along the duodenal or peptic ulcers, and sort of protects the stomach from being, I guess, eroded more or ulcerated even more. But the important thing to note is that it only activates at acidic conditions. So if you give them an antacid to reduce the acidity of the stomach, it will kind of work against the sucralfate and make it, I guess, ineffective. The way I remember it is sugar sounds like, suk sounds like sugar, which sounds sticky. So it's a sticky paste. I don't know how good that is. And then I don't know how good this one will be, but sugar sounds like cake. And then you use aluminium foil to bake your cake. But I realized you actually use baking paper like a week ago. But until then, like aluminium for cakes, like go with that. That might help you. But you get aluminium toxicity as your adverse effect. Yeah, it works. The system works. All right, keep moving. Misoprostol. So this is a prostaglandin analog. So prostaglandins are important for inhibiting our gastric acid secretion, right? So if we have NSAIDs, if we block our prostaglandin production, we're going to get increased gastric acid secretion. And that's why NSAIDs are sort of associated with gastric ulcers. And so we need to kind of replace those prostaglandins in order to restore that sort of function of prostaglandins. So we use prostaglandin analogs in that aspect. So misoprostol is a prostaglandin analog. We use it in NSAID-induced peptic ulcers, sort of reverse the adverse consequences of NSAIDs. We can't use it in contraindications because prostaglandins they're also used, they're also a sort of biological hormone that induces uterine contractions. So you can get sort of early pregnancies, which is bad for babies. The way I remember it is pros sounds like prostaglandin, and then prost also sounds like prostitute. Prostitutes get pregnant, so don't use them in pregnancies. Yeah, we'll keep moving because that one's a bit more questionable as well. Bismuth chelate. So this one we don't really use, but again, it's sort of a protective agent on the ulcerated surfaces. Again, it only works at that acidic pH. And the adverse effects are black stools, black tongue. That's sort of the big thing to note here. Bismuth starts with B, black starts with B. They are associated. Just remember that. Like, you won't really need to remember more than that, I don't think. It's kind of like a sideline drug. It's not even, I don't think it's on the core curriculum either, on the core e-pharmacology either. Okay, antacids. So these are weak bases. They sort of, bases will reduce the acidity of the stomach, increase the pH actually. And there's two types you have. So you've got your aluminium hydroxide and magnesium hydroxide. Aluminium hydroxide causes constipation, whereas magnesium hydroxide causes diarrhea. I don't have a great mnemonic for remembering that, so you're going to have to try work that one out on your own. But yeah, just remember that they're weak bases, I think, and they counteract sort of that acidic function. Okay, so diarrhea, we talked about this a little bit before, but how we're going to treat our diarrhea. Most of it's self-resolving, and so we give our fluid and electrolyte replacements. But sometimes we might, if it's quite severe, we might need to give antibiotics or antidiarrheal agents, such as opioids or muscarinic antagonists. So the main fluid replacement therapy we give is oral rehydration solution. So this is your hydrolyte. So it's got a careful mix of sodium and water in it that sort of matches what your body is meant to have, the ratios that your body is meant to have. And that's why people also give them glucose, so sugary drinks, because sodium and glucose are co-transported. So both of them are there, it'll sort of increase the uptake into the cells and increase that rate of hydration. Yeah, so if it's sort of significant diarrhea and it's getting quite concerning, we might use loperamide. So loperamide, if we remember from our opioids last year, it's an opioid receptor agonist, so at mu receptors. So that's going to actually decrease our peristalsis and decrease our secretions, because it's sort of, it has that blocking effect on not only pain receptors in the central nervous system, but our mu receptors within the gastrointestinal system. And the reason it doesn't work as sort of an analgesic is because it's selective for the GIT. So it's not being absorbed into the bloodstream through the GIT. So it's just staying in the GIT, and it's working on that myenteric plexus instead of the central nervous system. So the main adverse effect is constipation, but we sort of use that to our advantage in terms of diarrhea. So if we're trying to basically constipate a person with diarrhea and not let the constipation go too far. Because if it goes too far, we get heavy constipation, we get toxic megacolon. So that's when you've got sort of this buildup of feces, and it sort of builds up, and your colon sort of dilates to the point where it's sort of non-functional, and it's just obstructed. And then your classical nausea, abdo pain, abdo discomfort, things like that. All right, constipation. How are we going to treat constipation? So what is constipation in general? So as opposed to diarrhea, it's decreased frequency of bowel movements or hardened stools. Features, difficulty passing if they're straining, if they've got anal fissures, if the stools are really, really hard, those are all signs of constipation. And there's a variety of different causes. I think one of the big ones to note are lack of dietary fiber. So if you're not eating enough wheat, things like oats and stuff like that, you might be constipated. Or you might have celiac disease. Celiac disease is one of the big ones as well, constipation. Yeah. So how are we going to treat our constipation? So we have a few different methods. So we've got purgative agents, so prokinetic agents. So these are going to make things move faster through our gastrointestinal tract. So bulk laxative, osmotic laxative, and stimulant laxatives, or our fecal softeners. So this is when things are sort of impacted and hardened. And we'll talk about each one of them. So bulk laxatives. So what are these? So they're poorly digested polysaccharides polymers. And because they're poorly digested, they sort of form this big dehydrated kind of mass, bulky mass in the GIT lumen. And because they're dehydrated, osmosis and osmolarity stuff, water moves from the large colon into the lumen. It moves along that osmotic ratio or whatever it's called. And that's why water sort of moves into the lumen and promotes that peristalsis. And the brand name for remember that is Metamucil. So Metamucil is a bulk laxative. Yeah. Osmotic laxatives. So again, these are poorly absorbed solutes. So these are salts and sugars. So they're actually higher concentration items as opposed to dehydrated items. So they draw water through into the gut again. But again, you've got this adverse effect of dehydration coming through. It's used before endoscopies, and the brand names to remember are lactulose and Movicol. Yeah, if you see PEG, polyethylene glycol, it's an osmotic laxative. Yeah. Stimulant laxatives, so these are increasing your electrolyte secretion. So instead of actually drawing things in, they're just making instead of being sort of passive movement of water, this is sort of an active secretion by the intestinal lumen. And this is again going to increase peristalsis, increasing lubrication of your feces. Brand name to remember is Dulcolax. Not much else important there. Senna is also commonly used. And then your fecal softeners. So these are reducing the actual surface tension in the feces, and they have a weak laxative effect. So if people have really, really hard stools, if they're straining to like get them through because they're stuck basically in the colon, this is what you will use. Most old people have this because old people are old. And the brand name to remember here is Coloxal. So these are your fecal softeners. All right. Next one we're going to be talking about is vomiting. So in order to understand how we treat vomiting, I think it's important to note how vomiting works because there's a lot of different mechanisms. And it's not exactly the best understood concept in medicine either, because it's such a complex, it's like a psychogenic process as well as a chemical process. So we'll try and talk about it. So vomiting is stimulated from this area in the brain we call the chemoreceptor trigger zone. It's in the area prostrema. These are just buzzwords to remember. And I think it's not that important until you do neuro, to be honest. But the important thing to note is that it's located outside the blood-brain barrier. So we don't need our drugs to be specifically crossing the blood-brain barrier in order to target our chemoreceptor trigger zone, in order to target vomiting. So there's a bunch of different inputs that activate sort of our vomiting center in the chemoreceptor trigger zone, not only from the GIT, but we also get it from mainly the ear and your vestibular nuclei. So your hearing, alterations in hearing or your balance, that can also cause vomiting or stimulate vomiting. And the key neurotransmitters to remember are acetylcholine, histamine, serotonin, and dopamine. And these are all, I think it's better to learn them through the drugs that target them rather than the actual physiological mechanisms, because they're quite complicated physiological mechanisms and there's a lot of overlap between them. this is sort of a diagram, I guess, summarizing it all. But if you see in the chemoreceptor trigger zone, you've got your dopamine, you've got your serotonin, you've got NK1, which we'll also talk about. And this will all act on the vomiting centers in the medulla as well. So the medulla is stimulated by acetylcholine, again, your histamine, serotonin, and NK1, and opioid receptors. So we'll talk about each of them individually as we go through our pharmacological treatments. So these are the ones I just talked about. So there's certain indications for each of these, and specifically, in reality, there's actually a lot of overlap, and it's a lot harder to decide. But in terms of Monash buzzwords and what they want you to know, they kind of box them into different things. So muscarinics are for motion sickness, histamines are for motion sickness, but also morning sickness. Dopamine receptor agonists are when you've got contraindications, you can't use some of the other ones. Your serotonin antagonists are for your post-operative vomiting or for your chemo. And then NK1 is sort of this new emerging class of antiemetic agents. All right, so we'll talk about antihistamine first. So it targets the histamine receptors in the vestibular nuclei. So if we're blocking the input into the vestibular nuclei, we're blocking the input from the vestibular nuclei into the chemoreceptor trigger zone. And so we're blocking that stimulation of vomiting there. The main thing to note is that generation 1 antihistamines, there's 2 generations of antihistamines. So your generation 1, there's your generation 2. Generation 1 does cross the blood-brain barrier, and so it can affect all sort of parts of your body. Whereas your generation 2, it's more used for hay fever and sort of localized allergic reactions. So generation 1, I sort of imagine like 1 is like 1 with your entire body. Your body is 1, so that's why it targets your entire body. And the main indication here is for motion sickness. So if people are getting sick after like sitting in the car, going on airplanes, going on like boats and things like that, that's when you'll generally use antihistamine, promethazine, as a first-line sort of treatment. It's got sort of anticholinergic adverse effects as well to be noticed of. It's sort of due to a bit of overlap. They don't exactly know why, but it just is. So you've got your classical anticholinergic effects, so your blurred vision, mydriasis, I read blurred vision twice, dry mouth and urinary retention, and then sedation due to central effects of antihistamines as well. Hyoscine, so this is a muscarinic agonist. So this is again blocking muscarinic receptors. So again, you've got that anticholinergic adverse effects as well. So blurred vision, urinary retention, decreased salivation, drowsiness. If we remember back to atropine, it's similar. It rhymes with atropine. Atropine sounds like trees. You need big, strong muscles to cut down trees. And so it's muscarinic antagonist. That's the way I remember it. Yeah. Metoclopramide. So this one's a little bit hard to remember, but it's a dopamine antagonist. So you've got a lot of dopamine receptors which stimulate vomiting in the chemoreceptor trigger zone. So if you block those receptors, you're going to block your vomiting. The main contraindication to note here is that if people have Parkinson's disease, you don't give them this drug because Parkinson's disease is, I guess, characterized by a lack of dopamine. And so if you're reducing the effects of their dopamine already by blocking their receptors, it's going to sort of exacerbate their Parkinson's. And that's what you see in their adverse effect. It's sort of an exacerbation of Parkinson's disease. But again, more in neuro. Just note that it's a contraindication is Parkinson's disease for metoclopramide. Domperidone, so this isn't core, but they talk about it. So this is a peripherally selective dopamine receptor antagonist. So this is what we use if people do have Parkinson's disease, because it's not going to affect central Parkinson's receptors. But yeah, don't worry too much about that, I don't think. It'll help you later on though, I think. Ondansetron, so this is probably a big one. This is one of the most important ones actually used clinically and can be examined importantly. So this is it's a serotonin antagonist. working in the chemoreceptor trigger zone again. And the main indications are acute vasomesis. So if people just suddenly start vomiting and you really want to stop their vomiting, you give them ondansetron because it's quite effective. If they've got post-operative vomiting, or chemotherapy, if they've got post-chemo vomiting, because that's a common side effect of chemotherapy, you give them ondansetron. The way I remember it is ondansetron sort of sounds like alien-like. Cancer is sort of like alien-like, like it's a bit all like weird and wacky. So that's how I link it together. And like set sort of sounds like serotonin if you like rearrange the letters there. So that might help you remember that it's a serotonin antagonist. Aprepitant. So this again isn't core, but they talk about it a little in your lectures, I believe. So it's an NK1 receptor antagonist. So you've got this substance called substance P, which acts at NK1 receptors in the chemoreceptor trigger zone. And so if we block those, we're going to block vomiting. It's pretty effective. It's effective in a lot of different things. So chemo, motion sickness, and alcohol excess. But it's new. It's not really first line. They don't know all the side effects of it yet. So don't pick it. Just be aware that that could be an option they try to trick you with. And this is sort of a summary of everything that I've just talked about, where the different things are acting on, and how it all sort of comes together. Right. So that's a lot of sort of, I guess, didactic teaching that I think you're going to have to learn and just kind of rote learn. I think Pharm and Micro is kind of a little bit like that, which kind of sucks. But I always found that flashcards helps for pharmacology in particular, and using those mnemonics and like memory tools to help you is always a good thing. Yeah. Does anyone else have any great mnemonics that they use to help remember stuff? Or are mine just too brilliant?
No, you always have the best
wonderful.
one.
Yeah, we'll go with that.
All right. Are there any questions that anyone would like to ask?
Gabby or Elena, stop the recording.
Oh, yep.